Hello biologists, this is Miss R and we're going to talk about Lab 1.09, replicating DNA. The process of replication, of course, is copying DNA so you can make a brand new cell. The first thing we're going to do is look at this fabulous animation and look at some of what the different enzymes do. Enzymes, of course, are a particular kind of protein that catalyze reactions. In other words, they help reactions happen faster. There are a lot of reactions that go on in DNA replication, and we're going to look at those. Make sure you pay attention to these actors in the replication process. You'll also be able to check out this animation yourself later. Right now we're going to watch as a DNA strand here, you can see all the bases, GATC, gets copied. So you can see the first enzyme here is called helicase, and it's kind of like the zipper pull that unzips the zipper. Here is primase coming in. Primase helps match up the bases, and this red one is called primase. It helps get the process started so polymerase can come in. You can see how primase came in and got things started, and then polymerase came in and kept matching up these bases. This happens very, very quickly in your cells. Don't worry if you don't quite understand all of the different actors in the process. I'm not going to require you to know everything, but I just want you to get a big picture understanding of how everything works together to copy DNA. Let's keep rolling here. You can see that primase continues, and you can see that this bottom strand or lagging strand of DNA kind of goes in the opposite direction of the top strand. That's because DNA will only prime in one direction. And then some proofreading enzymes come in and kind of fix things up. And then there are a couple nicks left. And so this last actor in the process is called ligase, and it just zips up all those little nicks in the zipper. So let's review what we saw in the animation. Ligase kind of fixed the nicks at the end. Helicase was the zipper pull and unwound the DNA and split the two halves apart. The two polymerases added bases to the half of the DNA as it was opened up, and primase kind of got things started. It put a few bases in so polymerase could get started. It might be helpful to think about DNA as a zipper. There's two sides. There's a sugar phosphate backbone on each side, and there are bases that match up and kind of stick the DNA together. DNA replication happens, the zipper gets unzipped by helicase. That's the zipper pull. And then new DNA gets brought in, and you build basically the other half of the zipper piece by piece by piece with the enzymes. And here, when you're building a new DNA, the zipper is kind of a polymerase because it's pulling in those new pieces and adding them. Now you get to be DNA polymerase. We're going to go to an animation where you get to build some DNA. If you want to check this animation out on your own, you certainly can, and I encourage you to. If you put in the URL, you're going to click Build DNA. Then you can read the instructions, but I'll show you what to do right now. DNA has complementary base pairs. That means they have base pairs that always match up. In other words, A always goes with T, and G always goes with C. Or if G is already there, you bring in C. You can see the colors, like green always goes with yellow, and purple always goes with red. I'll put in a couple of bases. Then we need to build the backbone. That's kind of the outside part of the zipper of DNA. That's the sugar phosphate backbone. And we do that in this animation here by bringing in deoxyribose. That's the name of the sugar in DNA. DNA, of course, is deoxyribose nucleic acid. So deoxyribose, the sugar, is the D in DNA. And then we bring in a phosphate group which bonds the two sugars together. And this kind of makes the outside of the zipper. You can think of the bases as the teeth of the zipper. You can see like the base is here, and this is the sugar phosphate backbone, that purple and yellow thing that we're making. So hopefully you get the idea. You put a sugar phosphate backbone in place and you make these complementary base pairs. I could keep going, but I think you probably get the idea. And DNA also does this on the other side at the same time, but it starts from the other end. You can think of as DNA like reading. You only read left to right, and DNA only gets replicated in a certain direction. And then I'd add the sugar phosphate backbone here. And basically what we're making, now, we started out with one zipper, and we're going to end up with two complete new zippers. And so you get the idea of how you match up the base pairs and you add the sugar phosphate backbone. If you think of the black zipper as the original DNA, it got unzipped, and here we're making two new strands of DNA. Obviously there wouldn't be this part of the zipper hanging off in replication, but we're making two new separate strands, and each of the new strands is basically half the original strand and half new bases. Here's the other one. Half the original strand, half the original piece of DNA, and half new bases brought in by polymerase. Don't panic, but this is what polymerase really looks like. It's pretty complicated. Obviously, the animations we've been looking at have been pretty simple. And it's okay, you don't have to understand how this all works, but I just want to give you an idea of how beautiful and crazy and complicated our DNA is, and it gets replicated in your cells all the time, and you don't even have to think about it. So let's take a moment and think a little bit more about what we just did. We added complementary base pairs, so we knew that A always went with T, and G always went with C. What did we add right here? You can go back in the video if you need to review. Remember, we also added bases to the bottom strand. With T, we'd always match A. With C, we'd always match G. With G, we'd always match C, etc., etc. So you should be able to guess the order of the bases we added just from looking at what's there. Here's a picture of what the DNA looked like when it was all matched up. And I want you to notice what's in these blue boxes here. Notice the sequence here is the same. There's C and C, A and A, A and A, T and T. So these were the two strands that were original. We peeled open those strands and we added bases here that basically matched the old strand. See, the new strand here that we created matched the old strand. And that complementary nature, that means it's not the same. G doesn't match with G, G matches C. That complementary nature is what makes, is one of the things that helps DNA be copied with fidelity or copied with truth, copied accurately. We want to make sure our DNA is copied accurately because if things get out of order, then later when we try to make proteins from our DNA, they're going to be all messed up if things aren't in the same order when we copy it. Just like when you copy something, copy a piece of music, you don't want it to be all messed up because it will sound differently. We're going to learn a little bit more about how scientists discovered how DNA replicated. This is called the semi-conservative hypothesis, because when we make new DNA, we talked about here how we keep half of the original. Semi, of course, means half. So we keep half the original, and then we make half the molecule new nucleotides. And so when we make new DNA, half is new and half is old. The scientists discovered this by adding nitrogen that was a little bit heavier than the normal element nitrogen. It's an isotope. And then they grew it in the new heavier nitrogen to see what happened. And you can see this bold part means it's the heavier nitrogen that gets involved in the DNA. So let's watch how DNA replicates. So you get the new stuff. They are going to put it in a centrifuge, which spins it out, and the heavy DNA, the heavy stuff, falls to the bottom. That would be here, kind of down below in the test tube. If it was all light nitrogen, it would be here, but you know it's half and half, half new DNA and half old, half heavy nitrogen and half light nitrogen, because it's in the middle of the test tube. So the first generation, when we grow it in heavy nitrogen, is going to be in the middle. Now let's talk about the second generation of DNA. The older generation of DNA that's made out of one old and one new, or one, in this case where they're growing it in heavy nitrogen, one half heavy and one half light. These DNA molecules will unzip and make new molecules, primarily with light nitrogen. So let's see what happens when these molecules are centrifuged. What happens after they're centrifuged and you take the test tube out of the centrifuge, or out of the thing that spins and separates the heavy from the light, you find out that there's a light DNA band for the new stuff that we just made with the light nitrogen, and there's still that middle band we had for the first generation. So scientists figured out how DNA replicated by using some isotopes of nitrogen to make half of the DNA heavy and half of it light. So when you ground it up and you spun it in a test tube, part of it would go to the bottom and part of it would go to the top. And by looking at how much went to the bottom and how much went to the top, they figured out how DNA replicated. Let's put it all together now. Let's look at this diagram. And we know the cell cycle. We know that DNA replication is part of the cell cycle. The cell cycle is when you have a cell that's just sitting there, and it's a brand new cell, and it starts to grow, and it gets ready for DNA replication or DNA synthesis. This stage is called G1, and then it goes into S phase, see the S here, and that's DNA replication. It copies its DNA. Then it prepares for mitosis, or basically copying all the other parts of the cell, like the mitochondria and the other organelles. And then it goes through PMAT, or mitosis, prophase, metaphase, anaphase, telophase, that we studied last semester. And then it divides, and the new DNA that was copied goes into the new cell. So the question asks, what color is DNA replication? What color is the DNA replication stage? Well, it's right here. And about what % of the cell life cycle does the cell spend replicating its DNA? Well, if you kind of slice this up as a pie, there are 4 pieces, and DNA replication is one of the 4 pieces. Why is it important that DNA replicates before mitosis? Well, if you're going to make a new cell, that cell needs a full copy of all the DNA to be a new cell. Thanks so much for watching Lab 1.09. See you in class.